So they all started in the nucleus with the transcription of the DNA code into an RNA code with the help of RNA polymerase and a lot of different proteins which act as transcription factors. And then this RNA, if it's in eukaryotes, is going to be processed and it's going to be adding a poly-A tail, a G-cap, and the removal of the introns to make an exon-only mature RNA. That's going to be happening with the help of spliceosomes and ligase. Then it leaves to the nuclear pores and immediately goes into the cytoplasm where enzymes are looking to destroy it. But if they don't get destroyed, a ribosome will attach themselves to it using the energy of the G-cap and a lot of initiation factors and help connect the smaller ribosomal unit and the large ribosomal unit and the first transfer RNA carrying methionine to initiate the translation process. As the ribosome will travel through the messenger RNA through elongation, and transfer RNAs will move from P site to E site and from A site to P site as another set of enzymes called elongation factors help these attachments take place, and an enzyme called peptidotransferase will actually help the polypeptide chain elongate over time. Remember, all of this will be controlled by the pairing of the codon with the anticodon with the help of elongation factors, and the transfer RNA is going to be carrying the proper amino acid. And it's carrying the proper amino acid because amino acid tRNA synthetase made sure that it was paired up correctly. And remember, those amino acids come from digestive processes that happen inside the cell. After the time when you reach the actual anticodon that sends the message to stop, this will also all end as the ribosome will receive the message of a release factor to release this and actually complete the protein synthesis process, and all the pieces will separate and the protein is done. And you can do this sequentially in an assembly line format in something that's called a polyribosome. That's pretty much an overview of what we talked about in protein synthesis. But after you do all of this, you're not yet done, especially if you're in a eukaryotic cell. Proteins can still be changed in a lot of ways. You can make changes to the structure of the protein. You can make folding of the structure of the protein. You can cut the protein into smaller chunks. You can make the protein with small chunks get together to make bigger chunks. Remember, subunits sometimes get together to make a quaternary structure. You can fold the protein in different ways. And so there's a lot of things that can be done. You can methylate the protein, you can phosphorylate the protein, you can charge the protein, you can add sugar to the protein, you can add fat to the protein. There's a lot of different things that you can do to change the structure of the protein before the actual function is performed, whether inside or outside the cell. And so, protein synthesis is not done at the end of actual translation, but you also have protein processing. Now, something that will happen during protein processing, for example, is some proteins will actually be misfolded and therefore will need to be destroyed and start over. Other proteins are going to be sent to work on specialized clients or specialized purposes, and they will have to be changed. Other proteins are going to be more generalistic challenges, and they're not going to have to necessarily to be changed. Doing those changes, though, it's possible that they will be misfolded again, and therefore be degraded, and therefore restart the process. Or they're going to be refolded into actual useful proteins again. Also, some proteins are going to be sent to export outside the cell, and they must be translocated into the rough ER, so they're going to be packaged to delivery outside of the cell with the help of the rough ER and the Golgi apparatus. Lots of different things need to happen to these proteins. This whole thing is coordinated through several different enzymes. Again, proteins help make proteins, so it's ironic. You need proteins to make all this happen. The beginning of the protein, remember from the beginning of this lecture series, is always going to be called the N-terminus. And that's the beginning because it has the first amino acid, which starts with an amino group. Right after the N-terminus, you're going to have a sequence of amino acids, which usually serves as a signal sequence. This signal sequence is a specific sequence of amino acids which tell the cell whether this protein is going to be used inside or outside of the cell. If the signal sequence says this is a protein for the outside because it has this specific sequence of amino acids right at the beginning, this means this protein needs to go inside of the rough ER and then into the Golgi and then into a vesicle and then sent out through exocytosis. That means this protein needs to be incorporated into the endomembrane system. How do you do that? Well, a special protein plus RNA molecule called small recognition particle, or SRP for short. And again, this is made of a combination of protein and RNA, and this is the sixth use for RNA in the cell. You get RNA, which is part of SRP particles, called SRP RNA. Will serve as a piece that's going to connect itself to the signal sequence of the protein that was just made. And then is going to take this protein and attach itself through an SRP receptor at the surface of the endoplasmic reticulum. Then the ribosome will start working with this protein and continue working with it, but the protein is going to be made in a way that it's going to be thrown into the lumen of the rough ER, or the inside of the rough ER, even as it's made. And so as the protein is completed, it's going to be trapped inside of the rough ER and be dragged within the rough ER and changed and folded within the rough ER and then sent into the Golgi and then sent out of the cell through exocytosis in a vesicle. And so it's very important for these signal recognition particles, or SRPs, to be looking for proteins that have sequences which tell the body you need to send this outside of the cell, and so they will connect to the signal receptor particle in the membrane of the rough ER, and then the protein is going to be put in through a pore into the rough ER and be trapped there for exportation from the cell. Other classes of proteins also help proteins in different ways. There's a special kind of protein or enzyme called chaperone proteins, which help proteins fold in the appropriate way. Once you finish the polypeptide chain, you still have to create secondary, tertiary, and sometimes quaternary structure, which means protein needs to be folded correctly. Without the help of chaperone proteins, it's almost impossible to make that happen correctly. And therefore, sometimes proteins will fold incorrectly and will need to be refolded or discarded in actual process which digest the protein, which we talked about earlier in the lecture series, which includes the action of lysosomes, and sometimes they specialize in destroying proteins, and they're called proteasomes, and also of free enzymes in the cytoplasm that we call proteases. So structural changes to the protein will actually take place, and the formation of tertiary quaternary structure of the protein is going to all have to do with the chaperone proteins, which are actually going to do this. And you're also going to have chemical changes to the protein. You're going to have proteins that are going to be clipped into smaller chunks, and some parts of it are not going to be necessary, they're going to be thrown out. Other proteins are going to be split into two, and you actually made two proteins out of one. Other proteins are actually becoming made of putting together different kind of subunits to create different domains and different functions in this protein. And that whole split gene theory that we talked about has everything to do with this. And so a lot of things will be happening to the protein after the process is actually completed. As an example, insulin, which gets sent out of the cell, would actually be made by a ribosome which will attach itself to the surface of the rough ER, because as soon as the ribosome makes the signal sequence, or the first few amino acids, an SRP molecule recognizes that as a protein destined for export of the cell, and therefore will drag that to connect to the SRP recognition particle on the membrane of the rough ER, and then the protein is going to be made through the pore of the rough ER and then trapped inside the rough ER, when it's going to be folded, oxidized. The signal protein sequence is actually going to be cleaved, so you see like how you can remove the extra stuff, and then the protein is going to go through the rough ER for export to the Golgi, packaging to a vesicle, and then sent out of the cell through that vesicle. But within the Golgi, you're also going to cleave the protein to release some extra peptides which are not necessary, and you're also going to add other peptides which are necessary to produce the mature insulin. So if you don't do all of this process, you make an insulin which is not the right insulin. And so you see how you need to do all of this to actually create and process the protein. So protein synthesis in a nutshell involves the process of transcription, RNA processing, movement into the cytoplasm where it's going to be translated, and then finally modified after translation. And all of this is going to involve several enzymes, several proteins, elongation factors, transcription factors, termination factors, messenger RNA, transfer RNA, ribosomes, and lots of different players in the whole process. And it's a beautiful process that happens microscopically inside the cell. Human factors could learn something by this assembly line, couldn't they? But at each step of the way, it's an opportunity to stop protein synthesis from happening. And that sends us into the gene expression topic, where we talk about all the different ways in which protein synthesis can be controlled or regulated by stopping protein synthesis at any one of these steps that we talked about.